continuing on the theme of pioneers and pioneering developments for the pathways, the technology development pathways for medical nanobots. Our next speaker is Dr. Sonia Contreras. And Dr. Contera is visiting the conference from Oxford University's physics department, so a second speaker who has traveled overseas to attend the conference, and we really appreciate, Dr. Contera, your travel to the conference. The topic is certainly one which is a fundamental pathway for medical nanobots, nanotubes, with the title of the presentation being Nanotubes as a Drug Delivery System. Please join me in giving a warm welcome to Dr. Sonia Contera.
Thank you for inviting me. I'm very jet lagged. I haven't slept a single hour last night, so I hope you will understand if I'm not very good today. Right. Coming from the University of Oxford, we have a new I've been running for a few years, Bio-Nanotechnology Center. And what I've been worried, I'm a physicist, not an MD, as it was written before. And what I've been worried in the last 10 years of my life is to find ways to look at disease or biological function at the nanoscale in physiological conditions. This is not easy. It's not easy to look at a protein functioning in physiological conditions and see it at the nanoscale. So this has taken me to develop techniques, to improve techniques that they were already developed, such as a scanning probe microscopy, atomic force microscopy, and recently has led me to use nanoparticles, and in particular carbon nanotubes, for studying at the nanoscale biological function. In the last 2 years, James Martin, which is our benefactor, liked our ideas and gave us a big grant, and we're starting to use this or trying to understand what are the interactions of nanoparticles at the very basic level with cells and with proteins and DNA. So to start with, I put you one of the my slide, one of the pictures of one of our atomic force microscope, where you can see an E. coli cell that is dividing. You can see the DNA, and I've put cartoons of nanotubes so you can see the sizes where we're working. Right. So let me tell you where carbon nanotubes, in case some of you don't know it. So a few years ago, Ijima in Japan found these carbon structures when they were looking for other things, and they saw the growth of these very thin tubes of carbon. These very thin tubes of carbon that come in two ways. Come in two ways: single-wall carbon nanotubes and multi-wall carbon nanotubes. The idea of them is very simple. It's a carbon sheet that it folds on itself and forms a tube. However, there are many ways that you can fold this tube. It has a chirality. And the physical characteristics of these nanotubes will depend on this folding of the carbon sheet. We also have multi-wall carbon nanotubes in which different nanotubes are growing concentrically to form a bigger nanostructure. Now, why are we interested in single-wall carbon nanotubes at all for nanomedicine? Well, single-wall carbon nanotubes, as soon as they started, the physicists like me thought, oh, this is great. This is a one-dimensional system. We can learn a lot of things about solid-state physics and condensed matter physics that we didn't know before. And what they found with all these studies are really interesting characteristics for us that are trying to treat diseases, and we're trying to understand biological function at the nanoscale. I have summarized some of them here. So we have a strong optical absorption in the near infrared. I think the talk after me will be talking more about this, but this means absorbing the infrared. So in principle, you can target a tumor. You could go there with your nanotube and just blast it with heat from outside. It will heat, and this has been proved already that this can be done. From a physicist's point of view that is trying to image biological function in the cell, these semiconductor and single-wall nanotubes have photoluminescence, means fluorescence, in the near-infrared range. So we can put a marker inside the cell that in principle we could detect by near-infrared fluorescence. Plus, they have a very good resonance-enhanced Raman signal, so we can see them moving inside cells. Right. The single-wall carbon nanotubes have very interesting physical characteristics that are very good for detecting them once you put them in a body. But for reasons I will tell later, they're difficult to deal with, to make them interact with biological systems in a good way. We also have multi-wall carbon nanotubes, which are larger in size. And therefore, due to this size, because size matters at the nanoscale, we can tune the size, the diameter, and the length, so we can make them interact with lipid bilayers, with DNAs, et cetera, et cetera. I will show you some examples we're doing in the lab later. Right. So let me tell you what is the problem then. We're trying to deliver drugs to cells using carbon nanotubes. And you see the problem we have. We're injecting, for example, particles in the body, and we need them to reach the tumor. We need them to reach the place we want. So we need some antibodies or whatever proteins attached to the surface of the nanotube that will target that specific area of the body. As you have seen in the previous talk, it's not easy to make proteins stick to a structure, let alone a nanostructure, its functionality. Then we need them to circulate for a long time in the blood, long enough so they don't stick around and then they don't reach your tumor. We need, importantly, to cross the cell membrane, and this is not a trivial problem. And then we need to identify a vital cellular process that we want to interact with. I have put you here the cartoons of eukaryotic cells, prokaryotic cells. We're working with both. We're trying to interfere with several processes within the cells I will show you later. And specifically, one of the main efforts that we're doing at our Institute of Nanoscience for Medicine is to study at a very basic level the interaction of carbon nanotubes with membranes. First, because the membrane, as it has been shown before, can be used for making vesicles that then can be put into a body, and also because we want to traverse this membrane and we need to understand how we do this. So, as I said, there is a growing controversy in this field that one of the main problems we're having is that what are these nanostructures going to be and how can we control them? How can we control their action in the body? Are we talking about the difficulty between distinguishing between nanomedicine and nanotoxins? So the main goal of my institute and my lab in this research is to understand how the basic processes, the interactions with DNA, proteins, membrane, and ultimately with cells, we want to understand the basic science so we can understand the science, use it for drug delivery, and avoid nanotoxicological effect. So basically what I'm going to talk to you and on my drive is to understand what happens on the molecular nano level. Now, just to show off some of the beautiful images we get in my lab with these new imaging techniques I was telling you about. This is, and also for setting you visually what is the problem we have here. This is, for example, let's change the color. This is one of the atomic force microscopy image of my group where we can see bacteriodopsin. It was talked about before. It's a model system for GPCRs in solution, and we can reach a resolution. I don't know why the brightness of the screen went up because we lost some. Okay. So we can see that now, with the techniques we're developing now, which are also nanotechniques, of course, we can reach to see a single membrane protein with a resolution of 3 angstroms without the need of a crystal at room temperature and in solution. I'll show you here also some potassium channels that we are studying now. And importantly, I'll show you here the molecule of DNA that now we can image with a resolution that we can see the double helix in solution. This is a supercoiled DNA extract from a bacterium. This is real DNA. And what we're trying to do when we're interacting, when we throw carbon nanotubes to a cell, this more or less the size, is to interact with all this range of biological machines. Right. So What I'm interested is in the mechanisms. There's several groups that have thrown, more or less crudely or less crudely, carbon nanotubes to cells, and they have looked at if does the cell take it in or what does it do with them. So all the results are quite controversial. Some cells will swallow these nanotubes by endocytosis, or sometimes the tube just goes in by diffusion because of these interactions of the nanotubes with the cell membrane, but nobody really knows what is happening in a cell. Depends on the cell, depends on the tube, depends on the surface of the tube. So we're trying to do a more systematic approach and try to really understand, can we use these as drug delivery systems or for antibiotic cargoes? We still don't know. There's a long way to go because we don't know the basic science. We don't understand how the cell membrane works. We don't understand how a nanoparticle will interact with the cell membrane. So there are many, many problems. So we can actually bring a nanorobot. Okay. So I'll go quickly now. Okay. So there's many ways I was telling you before that people have been trying to approach the problems, sticking all sorts of things to the carbon nanotube surface. Lots of the studies of, as I was telling you before, of toxicity in mice, and they're all inconclusive. Some kill the mice because they actually feed in them enormous amounts of nanotubes that I think we could, anybody, some are not toxic. So toxicity is controversial. What we know is that simple acid treatment, we have to, carbon nanotubes are extremely hydrophobic, and this is the main problem. So as I was showing you in this slide, they aggregate. That it's very difficult to make treatments of carbon nanotubes that don't make them aggregate in solution. So one of the main problems we're trying to overcome is to make soluble, biocompatible nanotubes that are not going to aggregate in the body and actually cause disease. So our approach has been doping the carbon nanotube with nitrogen. So we are introducing nitrogen atoms in the carbon structure. And in this way, through different mechanisms and acid, lots of chemical processes, we see that all these impurities put in the carbon nanotube surface give us different advantages. First, we can cut them to the desired size we want by a simple boiling them in acid, basically. So that's the carbon nanotube, and different acid treatments leave us to a position that we can cut the tube. And then, which is the nice thing for us, these carbon nanotubes that have been doped with nitrogen are good for coating with protein. These are nanotubes that are normal nanotubes without this nitrogen, and the proteins usually stick to the ends. We have not very good coverage, but when we do these acid-treated carbon nanotubes, we can coat them all along with proteins that are functional. We did several spectroscopies and different studies to show that these proteins actually are correctly folded and are functional. We have used these carbon nanotubes because these nitrogen doped carbon nanotubes have also very interesting electrical properties. So we wanted to build biosensors within, as they were saying before, you want to sense things inside the body. And one way of doing this would be with electric currents. So we have to study first from the physics of the nanotube, then move on to what happens when you attach proteins to the nanotube, and how are the changes in the conductivity of the devices we are making. And here, for example, we can see that we can detect antibody protein binding to carbon nanotube devices with very high sensitivity. As I said before, we're interested in the interaction of nanotubes with membranes. We're collaborating with a group doing molecular dynamic simulations and showing that actually membranes interact strongly with carbon nanotubes. As you can see here, which if you put in a simulation a nanotube through a membrane, it gets all coated. So what we're trying to do is not easy either, is to coat a carbon nanotube with a lipid bilayers, and we had to do a systematic study of how much has to be the diameter of the tube, because lipid membranes cannot bend infinitely. They will have a radius of curvature that they can coat. So we, for example, find out things like we need a nanotube at least to be 40 nanometers in diameter, so we can coat it with a lipid bilayer. We did all sorts of tests to see that it actually was coating, et cetera, et cetera. We have managed to put membrane proteins inside the lipid bilayer that coats a nanotube. That could be, again, a good platform for testing drugs, specifically targeting membrane protein. And I'm very lucky to work with Dr. Sonia, which is fantastic biologist and DNA physics. And talking about energy, as it was being talked before, we think that there are many DNA proteins that are running, DNA motors that are running around DNA. Our DNA inside the chromosomes is incredibly closely packed. So we're trying to understand how these proteins are running around this DNA and what are the diseases and how can we target a specific protein. So if, for example, we want to cure cancer with a nanoparticle. So we are studying DNA topoisomerases that change the topology of the DNA. And actually, one of our fundamental questions is that if the proteins are using the DNA in the cell is wound up and is storing a lot of elastic energy. And we think that DNA proteins are moving along DNA using this elastic energy. This is also something we're trying to study. So anyway, one of the main targets we are studying is these DNA topoisomerases that are fundamental because if your DNA in your cell has strange structures and have loops like that, you die. And DNA topoisomerases change the DNA structure and keeps it clean of knots because DNA has a topology. And this is something we're trying to understand and we're trying to learn and we're trying to target with carbon nanotubes. So this is the sort of this is the strategy we're trying to follow, target proteins such as DNA topoisomerases with carbon nanotubes, and try to interfere with molecular processes such as DNA replication, DNA transcription, and then try to kill cells in this way. So for doing that, we're going to use the physical properties of DNA in alive. So we're going to wind up DNA. This we can do in the lab. And Sonia can make all sorts of DNA shapes, like relaxed DNA, positive supercoil wound up in one way, negative supercoils wound up in the other way. And by doing this, we're hoping we can make it interact with single-word carbon nanotubes. The way we want to do it is one of the ways we think it will interact, is because negative supercoil DNA, which is the way in which most DNA in your cells will be present, will have areas in which a double strand is lost, and we have single-stranded areas that are known to interact strongly with DNA, with carbon nanotubes. So we have some experiments done with this. It's still we are not very convinced about it, but we're hoping that we can start inserting intercalants in the DNA that will be these carbon nanotubes. And from there, we will go on to see what we do with them. First, we can, for example, target proteins like topoisomerases for tumor cell, for targeting tumor cells. And then, of course, we're interested in the nanobot idea of can we use the elastic energy stored in DNA to actually move a nanoparticle along the DNA. Right. So with this, I finish. And thanking my collaborators, and especially thanking James Martin for funding all our research in nanotechnology.
Thank you, Dr. Contera. And we now have a few minutes for questions and answers. Please approach the microphone, introduce your name, and if you're comfortable, your affiliation.
Kester, Penn State College of Medicine, and Keystone Nano. Excellent talk. So you bring up the question of agglomeration with the nano.
Big problem.
Big problem. But the other problem is where the cargo actually goes.
Oh, yeah, that's.
For instance, you can bring the drug in, but it can be then pumped out by the ABC proteins. And or you want to get the DNA to the nucleus, but you may want to get sRNA to the cytosol. It's all ending up in the endosome most likely and ending up in the lysosome and being degraded. As you start looking at the different doping of phospholipids or nitrogen on the outside or size of these nanomaterials, do you see changes of where the cargo goes? Because that's what we really have to do down the road. How do we target, not to a cell, but where in the cell?
Yeah. And the problem is how you look at that. That's another main issue. We don't have tools to look at them. What I'm hoping is that the near-infrared photoluminescence of carbon nanotubes is going to help us a lot with that. So we are developing near-infrared microscopy to see if we can see where actually the nanotube goes. Because as it is now, even if the nanotube goes in, we don't know where it is.
And we're doing the same thing with near-infrared and calcium phosphate nanoparticles for the
That's
same
right.
type of, using the test tube, the cell as a test tube.
Yeah, that's right.
How much control do you have over the distribution of lengths of
The
the
lengths.
nanotubes? Yes, that you
The length
create.
is another problem because when you grow nanotubes, you usually grow them in these carpets and you can make them, they usually comes in microns. So one of the main problems is to cut them. It's not easy to cut them all to size. One of them, there's been many ways of doing this. We were successful with this nitrogen doping and then treating them with acid that we cut in those ways. The problem with nanotubes is sometimes they're so perfect that all you can do is etch them from the ends with acids. So that takes days and days of boiling them in acid to get them to the desired size. It's not easy. And also how to separate them in the right way is not easy. We're working with biochemical techniques to separate the carbon nanotubes, HPLC, sucrose readings. It's really not easy. It's really not easy.
So the other question I have, and now this is a question sort of out of ignorance because I'm not a nanotube person myself, but I'm thinking that I've heard of boron trinitride nanotubes that are more polar and therefore get around some of the issues of this extreme hydrophobicity.
These days, there's nanotubes made of all sorts of materials, silicon. The nice thing about carbon nanotubes, first, they're carbon, so they're not so toxic. And then there's so much physics being studied about them that we know much more about them, and maybe we can use them in better. They're easy to make as well.
Ruth Blumberg, NASA Astrobiology Institute. You raised the question of the potential toxicity, and it brought to mind the issues that came up with asbestosis.
That's exactly the same.
And the dimensions of these very small particles approximated that of DNA, essentially. Do you see some analogy there?
People have been looking for this analogy since the beginning. People have been feeding nanotubes to mice in different ways and different conditions. They don't seem to develop asbestosis. Only one case, but then again, it depends on the surface chemistry of the nanotube. Actually, they're surprisingly non-toxic. So of the tests I have done in the lab, in vitro, never in vivo, the cells love them. But then again, we don't know we need more studies. But so far, there's been some clustering of carbon nanotubes. So, but there's always this very hydrophobic carbon nanotubes. Where the carbon nanotube surface is treated so it's hydrophilic and sort of biocompatible, they usually don't aggregate. But we're still far away from knowing what is the toxicity of nanotubes.
One of the issues with the asbestos toxicity was that it took a long time for it to, years.
Yeah. So what we're trying to avoid is exactly that, to put something around that then is going to cause things like asbestosis. We're trying to run ahead the problems, which also can be a problem in itself if we highlight all the problems and we don't look for the advantages. So I think we have to find a good balance in this research.
Thanks for an excellent talk.
Thank you.
Hi, Dave Zhang from Caltech. I have two technical questions / comments. And so you showed very beautiful pictures of doped and undoped carbon nanotubes with proteins around them.
Yeah.
So in the case of the undoped ones, I was wondering how you actually got it to disperse in solution very well, because that's one of the problems our lab was having a lot.
Oh yeah, we've been working a lot with that. We do these acid treatments, and they're not easy to disperse. When you add protein to them, they disperse better. But carbon nanotubes, as they are, they're very hydrophobic.
So when you actually add the proteins, it's still in an acid solution?
No, of course not. You take, proteins don't survive sulfuric nitric acid solution. You take them out, lots of spin down, lots of resolubilization. It's hard work. Even when you know the protocol, I think needs the magic hand of a good student to get it working.
I see. And the other thing that I want to mention is that someone in our lab was having really good success using locked nucleic acids, LNAs, to bind to carbon nanotubes. So maybe that's something you might want to look into.
I think I have found something like 25, 30 recipes for doing this job. You have to find the one that works for you, I think.
All right, thank you.
Dr. Contera, thank you very much for excellent presentation. And there was no sign of jet lag whatsoever. So it was really terrific. And I also want to thank you because, like Dr. Travis, you did an excellent job of identifying a technology development pathway for medical nanobots. And I would not be surprised in the future to that we end up speaking of a Travis energy pathway as well as a Contera nitrogen or protein conjugation pathway for medical nanobots. Really well done, and congratulations to your lab.